Somewhere deep beneath the ocean's surface, far from any sunlight, a small fish drifts through absolute darkness. Then, without warning, the water around it erupts in a constellation of blue-green pinpricks, each one a living creature disturbed by the fish's movement. In a forest half a world away, rotting logs pulse with an eerie green glow, as if the wood itself has captured starlight. And on a summer evening, lights dance through the air in patterns as old as the trees themselves, flashing messages only their own kind can read. This is bioluminescence, the ancient ability of living things to make their own light. And tonight, we will explore the quiet science behind these living lanterns. Welcome to Bedtime Biology, where science meets serenity. Each night, we explore the quiet wonders of the natural world, calming your mind and sparking your curiosity. If you like these videos, please consider subscribing to the channel and commenting below if you have any feedback or suggestions for topics of future episodes. At its heart, bioluminescence is a chemical conversation between molecules, a reaction so efficient that it produces light with almost no heat. When you strike a match, most of the energy released becomes warmth, the kind you can feel on your skin. But when a firefly glows or a jellyfish pulses with light, nearly all the energy goes directly into photons. This cold light has fascinated scientists for centuries, and understanding how it works requires looking at two key players, luciferin and luciferase. Luciferin is the fuel, a molecule that, when combined with oxygen, releases energy. The name itself comes from the Latin word for light bearer, the same root that gives us words like luminous and illuminate. But luciferin is not a single chemical. It is a family of related compounds, and different organisms have evolved their own versions. The luciferin in a firefly's abdomen is chemically distinct from the luciferin in a jellyfish bell or a glowing mushroom. These molecules share a common function, but arrived at it through separate evolutionary paths, a phenomenon scientists call convergent evolution. It is as if nature discovered the same solution to the problem of making light multiple times in different lineages across millions of years. Luciferase is the catalyst, the enzyme that speeds up the reaction between luciferin and oxygen. Without luciferase, luciferin might still react, but far too slowly to produce visible light. The enzyme brings the molecules together in just the right orientation, lowering the energy needed for the reaction to occur. When luciferin is oxidized, it transforms into an excited state molecule, one with more energy than it can comfortably hold. As it returns to a stable form, the excess energy is released not as heat, but as a photon of light. The color of that light depends on the structure of the luciferin and the environment inside the enzyme. Small changes in the chemical surroundings can shift the wavelength, producing greens, blues, yellows, or even reds. The firefly system is one of the best studied. Firefly luciferin, also called D-luciferin, reacts in the presence of adenosine triphosphate, the energy currency of cells, along with magnesium ions and molecular oxygen. The firefly luciferase first activates the luciferin by attaching it to an adenosine monophosphate molecule. This forms a luciferin AMP complex, which then reacts with oxygen to create an excited oxyluciferin molecule. When this molecule relaxes back to its ground state, it emits a photon of yellow-green light, the familiar glow of a summer evening. The entire process is astonishingly efficient, converting up to 90% of the chemical energy into light. In the ocean, different chemistries prevail. Many marine organisms use a luciferin called coelenterasine, named after the coelenterates, the group that includes jellyfish and corals. Coelenterasine does not require activation by ATP. Instead, it reacts directly with oxygen when the right enzyme is present. The luciferases that use coelenterasine are not related to firefly luciferase at all. They belong to entirely different protein families, yet they accomplish the same task. This is another example of convergent evolution, where unrelated organisms independently evolved bioluminescence, using different molecular tools but achieving the same result. Some marine bacteria use yet another system. These bacteria, including species in the genus Vibrio, produce light using a luciferin derived from fatty acids. The bacterial luciferase is a flavoprotein, meaning it requires a helper molecule called flavin mononucleotide. The reaction also involves a long-chain aldehyde, and together, these components produce a steady blue-green glow. Bacterial bioluminescence is often used by larger organisms through symbiotic relationships, where the bacteria live inside specialized light organs and provide their host with the ability to glow. Then there are fungi. For years, the chemistry of fungal bioluminescence remained mysterious. Researchers knew that mushrooms like Armillaria malea, the honey mushroom, could produce a faint green light, but they did not know what luciferin the fungi were using. It was only recently that scientists identified the fungal luciferin as a molecule called 3-hydroxyhispidine, derived from a precursor called hispidine. Fungal luciferase oxidizes this luciferin to produce light, and remarkably, all known bioluminescent fungi appear to use the same chemical system. This suggests that bioluminescence evolved just once in the fungal kingdom, and has been passed down through millions of years. The fact that bioluminescence has evolved independently so many times, at least 40 separate times across the tree of life, speaks to its usefulness. In the ocean, where 3/4 of all animals may be capable of producing light, bioluminescence serves countless functions. It is used to attract mates, to lure prey, to confuse predators, to communicate with others of the same species, and to camouflage against the faint light filtering down from the surface. On land, where bioluminescence is rarer, it still appears in fireflies, glowworms, certain fungi, and a handful of other organisms. Each has found its own reason for making light in the darkness. On a warm summer night in North America, the air above a meadow begins to flicker. One by one, tiny lights appear, drifting upward from the grass. These are fireflies, beetles of the family Lampyridae, and they are engaged in one of nature's most visible courtship rituals. The light they produce comes from specialized organs in their abdomens, where luciferin and luciferase react in cells called photocytes. Oxygen is delivered through tiny tubes called tracheae, and the firefly can control the light by regulating how much oxygen reaches the light-producing cells. When oxygen flows in, the reaction proceeds and the firefly glows. When the supply is restricted, the light fades. But fireflies do not glow continuously. They flash, and the pattern of those flashes is a language. Different species have different flash patterns, coded into their behavior by evolution. The Big Dipper firefly, Photinus pyralis, is common across much of North America. The male flies in a distinctive J-shaped pattern, about a meter off the ground, flashing once every 5 seconds or so. The flash lasts about 1 second. A female, sitting in the grass or on a low leaf, watches for the right pattern. If she sees a male of her own species performing the correct display, she waits exactly 2 seconds after his flash ends, then produces her own half-second flash. The male sees her response, flies toward her, and the exchange continues until he finds her. If everything proceeds correctly, they mate. This specificity is crucial. There are over 2,000 species of fireflies worldwide, and many live in the same habitats. Without distinct flash patterns, males and females would have no way to recognize their own kind. The timing, duration, and rhythm of the flashes act as a species-specific code. Some fireflies flash rapidly, others slowly. Some produce long glows, others short bursts. Some flash in synchrony with hundreds of other individuals, creating waves of light that sweep through entire forests. In Southeast Asia, certain species gather in mangrove trees and flash in perfect unison, the entire tree pulsing like a single organism. Scientists believe this synchrony may make the display more visible to females, or it may help individual males stand out when they flash slightly out of phase with the group. Not all fireflies use light for mating. The larvae of many species are bioluminescent, even though they cannot reproduce. Firefly larvae live in the soil and leaf litter, where they hunt earthworms, snails, and other soft-bodied prey. The glow they produce is thought to be a warning signal, a way of advertising that they are unpalatable. Fireflies contain toxic compounds called luciferins, bitter-tasting steroids that make them distasteful to many predators. A bird or toad that tries to eat a glowing firefly larva quickly learns to associate the light with a bad taste. This is called aposematism, the use of bright colors or lights to warn predators of toxicity. The same principle is at work in poison dart frogs, which are brightly colored, and in monarch butterflies, which are orange and black. Interestingly, the toxins that protect fireflies were not originally linked to bioluminescence. Recent genetic studies suggest that bioluminescence evolved in beetles before the luciferins did. The original function of the glow may have been something else entirely, perhaps related to oxidative stress, since the reaction helps to consume excess oxygen. Only later, as luciferins evolved in some lineages, did the light take on a secondary role as a warning signal. In adults, the primary function of the flash is sexual communication, but even then, the light may still deter some predators. Some fireflies have taken the system a step further. Females of the genus Futurus have learned to mimic the flash patterns of females from other species. After a Futurus female has mated with a male of her own species, she begins flashing the signals of Futinus females. When a male Futinus approaches, expecting to find a mate, the Futurus female captures and eats him. This aggressive mimicry serves a specific purpose. Futurus fireflies cannot produce their own luciferins, but Futinus fireflies can. By eating Futinus males, the Futurus females acquire the defensive chemicals, which they then use to protect themselves and their eggs. It is a form of chemical theft carried out under the cover of false signals. Firefly bioluminescence is not just beautiful, it is also threatened. Light pollution from streetlights, buildings, and other human sources is disrupting firefly communication across much of the world. Fireflies rely on darkness to see each other's flashes. In areas with significant artificial light, males have difficulty finding females, and females may not respond to male signals at all. Studies have shown that even low levels of artificial illumination can reduce mating success. Some species are more affected than others, depending on when they are active and how bright their flashes are. Species that flash at dusk, when ambient light levels are still relatively high, may be more resilient. Those that flash later at night, in complete darkness, suffer more when artificial lights intrude. Habitat loss compounds the problem. Fireflies spend most of their lives as larvae, living in soil, leaf litter, or rotting wood. When forests are cleared, when wetlands are drained, when lawns are mowed too short or treated with pesticides, firefly larvae lose the habitats they need to survive. Adults live for only a few weeks, just long enough to mate and lay eggs. If the larvae cannot find suitable habitat, the population collapses. In many parts of the world, firefly populations are declining, their lights growing dimmer year by year. Far below the ocean's surface, in a realm where sunlight never penetrates, bioluminescence is not the exception. It is the rule. Scientists estimate that 76% of marine animals in the deep sea are capable of producing light. In the mesopelagic zone, the twilight waters between 200 and 1,000 meters deep, bioluminescence is the dominant form of communication. Here, the water is not completely dark. A faint glow from the surface filters down, creating a dim blue background. Against this backdrop, animals have evolved light-producing organs called photophores, which they use for camouflage, hunting, defense, and signaling. One of the most dramatic uses of bioluminescence is seen in the anglerfish. Female anglerfish, which are much larger than the males, possess a modified dorsal fin spine that extends forward over the mouth like a fishing rod. At the tip of this lure dangles a bioluminescent organ glowing with light produced by symbiotic bacteria. The bacteria belong to the genus Vibrio, or related groups, and they live inside the lure in a mutualistic relationship. The anglerfish provides the bacteria with nutrients and a safe environment. In return, the bacteria produce light. The glow attracts curious prey, small fish and crustaceans, that mistake the light for food or for something else worth investigating. When the prey comes close enough, the anglerfish opens its enormous mouth and swallows it whole. The attack happens in an instant, aided by the anglerfish's ability to expand its jaws and stomach to enormous size. Not all deep-sea fish use light to attract prey. Some use it to hunt more actively. Dragonfish of the family Stomidae possess photophores beneath their eyes that produce red light. Most deep-sea animals are sensitive only to blue and green wavelengths, the colors that penetrate farthest in seawater. Red light is absorbed quickly and is rarely seen in the deep ocean. By producing red light, dragonfish gain a secret advantage. They can illuminate their surroundings and see prey that cannot see them, like a predator using a night vision scope. The dragonfish eye contains special pigments, including chlorophyll, which allow them to detect red wavelengths. This gives them a private channel of light, invisible to most other creatures in the ocean. Lanternfish, members of the family Myctophidae, are among the most abundant vertebrates on Earth. There may be more lanternfish in the ocean than any other kind of fish. They are small, typically just a few centimeters long, and they live in the mesopelagic zone by day, rising toward the surface at night to feed. Their bodies are covered with photophores, arranged in species-specific patterns. These patterns serve multiple purposes. They help lanternfish recognize members of their own species, which is important for schooling and mating. They also provide camouflage through a technique called counter-illumination. Counter-illumination works by matching the light coming from above. When a predator looks up from below, it sees the underside of its prey silhouetted against the faint glow from the surface. This shadow makes the prey easy to spot. But if the prey can produce its own light on its underside, matching the intensity and color of the downwelling light, it can erase its shadow and effectively disappear. Lanternfish accomplish this with remarkable precision. They have photophores on their bellies that emit blue-green light, the same color as the ocean background. Some species can even adjust the intensity of their light to match changing conditions, such as the presence of moonlight or clouds. The mechanism is sophisticated. Lanternfish have light-sensitive cells that detect the brightness of the water above them. These cells send signals to the photophores, which adjust their output accordingly. The photophores themselves are complex structures with lenses, reflectors, and filters that shape and direct the light. Some even have pigmented screens that can be moved to block or reveal the light, like opening and closing shutters. The result is a dynamic system that can respond to the environment in real time. Counter-illumination is not unique to lanternfish. Many other deep-sea animals use the same strategy. Hatchetfish, which have extremely flattened bodies, possess ventral photophores arranged in precise rows. Certain squid have photophores on their undersides, and they too adjust the light to match the background. The Hawaiian bobtail squid, which lives in shallow coastal waters rather than the deep sea, uses bacterial bioluminescence for counter-illumination at night, matching the moonlight filtering through the water. In all these cases, the principle is the same. Produce light downward to erase your shadow. Bioluminescence is also used for defense. When attacked, many deep-sea animals release clouds of bioluminescent particles or fluids. The sudden burst of light can startle a predator, giving the prey a moment to escape. Some squid, instead of ejecting black ink as their shallow water relatives do, release bioluminescent mucus. The glowing cloud hangs in the water, confusing the predator, while the squid darts away into the darkness. Other species take this further. The deep-sea shrimp, Acanthophyra purpurea, ejects a stream of bioluminescent vomit when threatened, creating a glowing decoy that distracts predators. The vampire squid, Vampiroteuthis infernalis, which is neither a true squid nor an octopus, but something in between, uses bioluminescence in multiple ways. Its body is covered with photophores that it can flash on and off, creating dazzling displays that may confuse predators. When seriously threatened, it can also expel sticky, bioluminescent mucus from the tips of its arms. The mucus glows for several minutes, and the vampire squid can control when and where it releases this substance. By leaving a glowing trail, the squid may convince a predator that it is somewhere it is not, buying time to escape. Some deep-sea creatures even detach glowing body parts as a distraction. The deep-sea squid, Octopoteuthis delatron, can shed one of its bioluminescent arms when grabbed by a predator. The arm continues to glow and squirm, holding the predator's attention while the squid escapes. This is similar to how some lizards can drop their tails, but with the added advantage of light to make the decoy even more effective. Then there is the burglar alarm hypothesis, a defensive strategy where bioluminescence is used to attract secondary predators. Imagine a small copepod being eaten by a fish. As the copepod is crushed in the fish's jaws, it produces a bright flash of light. This flash does not save the copepod, but it may attract a larger predator that eats the fish. By drawing attention to its attacker, the copepod may protect others of its kind from being eaten. This hypothesis has been tested in laboratory experiments with dinoflagellates, which flash when disturbed, and the results support the idea. Predators exposed to flashing prey reduce their feeding rates, perhaps because the light makes them more vulnerable to their own predators. On a calm night near the coast, the ocean can seem to glow. Walk along the beach, and each footstep in the wet sand leaves a trail of blue-green sparkles. Wade into the water, and your legs are surrounded by swirls of light. Move your hand through the surf, and the water ignites with tiny points of luminescence, as if you have disturbed a constellation. This is the work of dinoflagellates, single-celled organisms that drift in the plankton and light up when mechanically disturbed. Dinoflagellates are protists, a diverse group of mostly single-celled eukaryotes. They have two flagella, whip-like structures that propel them through the water in a characteristic spinning motion. Some dinoflagellates are photosynthetic, using chloroplasts to convert sunlight into energy. Others are heterotrophic, feeding on bacteria, diatoms, and other small organisms. Many can do both, switching between photosynthesis and feeding depending on conditions. Bioluminescent dinoflagellates are found in all the world's oceans, and more than 18 genera are known to produce light. The bioluminescence in dinoflagellates is controlled by structures called scintillons, tiny organelles about half a micrometer in diameter, distributed throughout the cell. Each scintillon contains dinoflagellate luciferin, a chlorophyll-derived molecule, and dinoflagellate luciferase. The luciferin in dinoflagellates is chemically unrelated to the luciferin in fireflies or jellyfish, yet another example of convergent evolution. When the dinoflagellate is disturbed by water movement, a wave, a swimming fish, or even the beat of its own flagella, ion channels open in the scintillon membranes. Protons flood in, rapidly lowering the pH inside the scintillon. This triggers the luciferase to oxidize the luciferin, producing a brief flash of blue-green light. The flash lasts about 1/10 of a second and is remarkably bright for such a small organism. A single dinoflagellate can produce multiple flashes if stimulated repeatedly, but eventually it exhausts its supply of luciferin and must wait for the molecule to be regenerated. This regeneration happens during the day. Dinoflagellate bioluminescence is controlled by a circadian clock and only occurs at night. During the day, the cells break down the scintillons and stop producing luciferin. As evening approaches, the scintillons reassemble, and luciferin production ramps up, reaching a peak in the hours after sunset. When dinoflagellates bloom, their numbers can increase to millions of cells per litre of seawater. During the day, these blooms often discolour the water, turning it red, brown, or pink, depending on the species. This is the origin of the term red tide, though not all red tides are toxic, and not all are caused by dinoflagellates. At night, if the dinoflagellates are bioluminescent, the water comes alive with light. Every breaking wave glows blue-green. Every fish that swims through the bloom leaves a glowing trail. The effect is so dramatic that it has been recorded by explorers and sailors for centuries. One of the most common bioluminescent species is Noctiluca scintillans, whose name means glowing bright by night. Noctiluca is larger than most dinoflagellates, about 1 to 2 millimeters in diameter, large enough to see with the naked eye as tiny translucent spheres. It feeds on diatoms, copepod eggs and other plankton, capturing prey with a tentacle and drawing it into a cytostome, a primitive mouth. When disturbed, Noctiluca flashes brightly, and in a bloom, the cumulative effect of millions of individuals can make the ocean appear to burn with cold fire. The function of bioluminescence in dinoflagellates is still debated, but the burglar alarm hypothesis is widely accepted. When a copepod or other small grazer tries to eat a dinoflagellate, the mechanical stress triggers a flash. The flash does not harm the predator directly, but it makes the predator more visible to fish and other larger predators. Laboratory experiments have shown that copepods reduce their grazing rates when exposed to bioluminescent dinoflagellates, supporting the idea that the light serves as a defense. Dinoflagellate blooms are influenced by a variety of environmental factors, including temperature, nutrient availability, and water circulation. In some locations, blooms occur predictably. The bioluminescent bays of Puerto Rico, including Mosquito Bay on the island of Vieques, experience persistent bioluminescence year-round due to the enclosed nature of the bays and the warm, nutrient-rich water. Kayaking through these bays at night is an otherworldly experience. Every paddle stroke creates an explosion of light, and fish darting through the water leave glowing streaks behind them. Not all dinoflagellate blooms are harmless. Some species produce toxins that can accumulate in shellfish, making them dangerous for humans to eat. These toxic red tides can cause paralytic shellfish poisoning, neurotoxic shellfish poisoning, or other illnesses. The toxins interfere with nerve function, and in severe cases, they can be fatal. Coastal communities monitor for red tides, closing shellfish harvests when dangerous species are present. The bioluminescence itself is not toxic, it is just light, but the organisms producing it may carry other, more dangerous chemicals. Interestingly, not all populations of Noctiluca scintillans are bioluminescent. Along the west coast of the United States, blooms of Noctiluca occur regularly, but they do not glow. Genetic studies have shown that these populations have lost the genes for producing luciferin, and in some cases, the luciferase gene has been silenced. This suggests that bioluminescence, while useful in some environments, may not be worth the energy cost in others, and natural selection can favor populations that stop producing light. In the summer of 1961, a young researcher named Osamu Shimomura traveled to Friday Harbor Laboratories in Washington State with a singular goal: to understand what made the crystal jelly, Aquaria victoria, glow. Shimomura had been studying bioluminescence for years, working with organisms like the sea firefly, and he knew that the glow in jellyfish came from a chemical reaction. But isolating the molecules responsible was proving difficult. He and his team collected tens of thousands of jellyfish, cutting away the bioluminescent tissues along the bell margin and processing them to extract the light-producing substance. The work was painstaking. After isolating a protein that seemed to be involved, Shimomura found that it produced only a faint glow in his laboratory tests. Frustrated, he cleaned up for the day and poured the remaining sample into a sink. The moment the liquid touched the seawater in the drain, there was a sudden, bright blue flash. Shimomura quickly realized what had happened. The calcium ions in the seawater were triggering the light-producing reaction. He had discovered aquarin, a photoprotein that glows blue when it binds calcium. But there was a mystery. Aquarin glowed blue, yet the jellyfish itself appeared green. Shimomura suspected there was another compound involved, something that absorbed the blue light and re-emitted it as green. He was right. Further purification revealed a second protein, one that did not produce light on its own, but fluoresced bright green when exposed to blue light. Shimomura called it green fluorescent protein, or GFP. At the time, it seemed like an interesting footnote to the story of equorin. Nobody could have predicted what would happen next. For decades, GFP remained obscure, a curiosity known only to a handful of researchers. Then, in the mid-1980s, a molecular biologist named Martin Chalfie attended a seminar where GFP was mentioned. Chalfie was studying the transparent roundworm Canohabditis elegans, trying to understand how genes were expressed in different parts of the worm's body. The problem was that the worm was so small and transparent that it was hard to see individual cells. Chalfie had a sudden insight. If he could insert the gene for GFP into the worm's DNA, the protein would glow wherever the gene was active, making it visible under a microscope. The idea worked. Chalfie and his team successfully expressed GFP in C. elegans, and the worm glowed green in exactly the cells they expected. This was a breakthrough. For the first time, scientists could watch where and when genes were turned on inside a living organism in real time, without killing or fixing the tissue. GFP became a marker, a biological flashlight that revealed the inner workings of cells. Other researchers quickly adopted the technique. Roger Tsien, a biochemist at the University of California, San Diego, began engineering variants of GFP that glowed in different colors. He created proteins that emitted cyan, yellow, and red light, each one a modification of the original GFP structure. With these multicolor variants, scientists could label different proteins in the same cell and watch them interact. The applications were endless. Researchers used GFP to track the development of neurons, to study the spread of infections, to visualize how cancer cells metastasize, to monitor gene therapy in living animals. In 2008, Shimomura, Chalfie, and Tsien shared the Nobel Prize in Chemistry for their work on GFP. The discovery had transformed biology. It had started with a simple question about why jellyfish glow, and led to a tool that illuminated the hidden processes of life itself. Equirria victoria, a small, translucent jellyfish that lives off the coast of the Pacific Northwest, had given science one of its most powerful instruments. The jellyfish itself uses GFP as part of its bioluminescent system. Equorin produces blue light when calcium ions are present, but the blue light is absorbed by GFP, which then fluoresces green. This process is called Förster resonance energy transfer, or FRET, and it shifts the wavelength of the emitted light to green, which may travel farther in seawater or be more visible to other jellyfish. The exact function of the bioluminescence in Equirria is not entirely clear. It may serve to deter predators, to attract prey, or to communicate with other jellyfish. What is clear is that this humble organism, collected by the tens of thousands for a single experiment, has changed the way we understand life. GFP is not unique to Equirria. Similar fluorescent proteins have been found in other marine organisms, including corals, sea anemones, and even some fish. Each variant has slightly different properties, and researchers have isolated and characterized dozens of them. These proteins form the basis of a whole toolkit for imaging and labeling in biology. Today, you can walk into almost any molecular biology laboratory in the world and find researchers using GFP or one of its relatives, to study everything from the behavior of individual molecules to the development of entire organisms. It is a legacy that began with a jellyfish glowing in the dark waters of Puget Sound. Walk through a forest at night, and you might notice a faint glow emanating from a rotting log. At first, it looks like a trick of the eyes, a phantom light that disappears when you look directly at it. But if you let your vision adjust to the darkness, the glow becomes unmistakable. The wood itself seems to be illuminated from within, pulsing with a soft, greenish light. This is foxfire, the bioluminescence of fungi, and it has been known to humans for centuries. The name foxfire is thought to come from the French feu follet, meaning foolish fire, though the exact origin is uncertain. In various cultures, the phenomenon has been attributed to spirits, fairies, or supernatural forces. In reality, it is the work of mycelium, the thread-like network of fungal cells that spread through decaying wood. Certain species of fungi in the phylum Basidiomycota are capable of bioluminescence, and the mycelium of these species glows continuously as it breaks down lignin, the tough polymer that gives wood its structure. At least 122 species of bioluminescent fungi are known, distributed across 4 major evolutionary lineages. The most widespread is the Armillaria lineage, which includes the honey mushroom Armillaria malea. Armillaria is a parasite and decomposer, attacking the roots of trees and spreading through forests in vast underground networks. In some cases, a single individual can cover many hectares, making it one of the largest organisms on Earth. The mycelium of Armillaria glows with a faint green light, but the fruiting bodies, the mushrooms that appear above ground, do not. Other bioluminescent fungi produce glowing mushrooms. Mycena chlorophyllus, found in tropical and subtropical regions of Asia and Australia, is one of the brightest. The caps and stems of this small mushroom emit a vivid green light, bright enough to read by if you have several clustered together. The glow is strongest in fresh specimens and fades as the mushroom ages. In Japan, these mushrooms are sometimes called hotaruteg, or firefly mushrooms, and they have inspired local folklore and festivals. Panellus stypticus, a small mushroom that grows on decaying hardwood, is found in North America, Europe, and Asia. Interestingly, not all populations of Panellus stypticus are bioluminescent. In North America, the mushrooms glow, but in Europe, they do not. This suggests that the bioluminescent trait has been lost in some populations, perhaps because the energy cost of producing light outweighs the benefit in certain environments. The chemistry of fungal bioluminescence was a mystery for many years, but recent studies have identified the key molecules. Fungal luciferin is a compound called 3-hydroxyhispidin, derived from hispidin, a molecule involved in the breakdown of lignin. The enzyme that catalyzes the light-producing reaction is a luciferase specific to fungi, and it oxidizes 3-hydroxyhispidin in the presence of oxygen, releasing green light. All known bioluminescent fungi appear to use the same luciferin and luciferase, suggesting that the trait evolved once in their common ancestor and has been inherited ever since. But why do fungi glow? The function of bioluminescence in fungi is still debated. One hypothesis is that the light attracts insects, which then help disperse fungal spores. Many bioluminescent mushrooms grow in dark, humid environments where they might otherwise go unnoticed by animals. By glowing at night, they could draw the attention of nocturnal insects, which land on the mushroom pick up spores on their bodies and carry them to new locations. This has been tested experimentally with artificial lights, and insects do show a preference for glowing objects in dark forests, supporting the idea. Another hypothesis suggests that bioluminescence is a byproduct of lignin degradation. The reaction that produces light involves molecules that are also used in breaking down wood. It is possible that the light itself serves no direct function, but is simply a side effect of the metabolic processes that allow the fungus to extract nutrients from dead trees. If that is the case, the glow is an accident, though a beautiful one. In some fungi, only the mycelium glows, not the mushrooms. This is true for Armillaria and related species. In others, like Mycena and Omphallotus, the mushrooms themselves produce light. In Mycena, Lamprospora, it is not the mushroom or the mycelium that glows, but the mature spores. Each evolutionary lineage seems to have found its own way of incorporating bioluminescence into its life cycle. Foxfire has practical uses in human history. Scandinavian travellers reportedly used glowing bark as trail markers, placing pieces of rotting wood along their path so they could find their way back in the dark. During World War II, soldiers in the Pacific Theatre are said to have used foxfire to read maps at night without giving away their position with electric lights. These stories may be exaggerated, but they reflect the fact that fungal bioluminescence, though faint, is real and can be surprisingly bright in complete darkness. To see foxfire yourself, you need fresh specimens, a dark environment, and patience. Bioluminescent fungi are most active during warm, humid conditions. Look for rotting logs in forests, particularly oak, beech, or other hardwoods. If you find mushrooms or mycelium of the right species, take them to a completely dark room and let your eyes adjust for at least 15 minutes. The glow will be subtle, usually green or blue-green, and it may be easier to see with peripheral vision. The experience is worth the effort, a glimpse into one of the quieter wonders of the living world. In the shallow coastal waters of Hawaii, a small squid emerges from the sand as the sun sets. It is no larger than your thumb, with a rounded mantle and large eyes. As it begins to swim, searching for prey, something remarkable happens. The underside of its body begins to glow with a soft blue-green light, matching the moonlight filtering down from the surface. This is the Hawaiian bobtail squid, Euprymna scolops, and the light it produces does not come from its own cells. It comes from bacteria. Inside the squid's mantle cavity, there is a specialized organ called the light organ. This structure is similar in many ways to an eye, but instead of gathering light, it emits it. The light organ contains chambers filled with bioluminescent bacteria of the species Vibrio fischeri. These bacteria produce light through the same basic mechanism as other bioluminescent organisms, using a luciferin derived from fatty acids and a bacterial luciferase. The reaction requires oxygen and flavin mononucleotide, and it produces a steady blue-green glow. The squid controls the light organ with remarkable precision. The organ is surrounded by reflective tissue on the top and sides, which directs all the light downward. There is a lens on the bottom surface that diffuses the light, making it appear uniform rather than coming from a single point. There is an iris, much like the iris of an eye, that can expand or contract to control how much light is emitted. And there are yellow filters that can be moved in front of the light, changing the color balance of the glow. The squid uses this light for counter-illumination, the same strategy employed by many deep-sea fish. During the day, the squid buries itself in the sand, where it is safe from predators. At night, it emerges to hunt small shrimp and other prey in the water column. As it swims, the light from the moon and stars filters down through the water. A predator, looking up from below, would normally see the squid's silhouette, a dark shape blocking the light. But the squid matches the intensity and color of the moonlight with its own bioluminescence, erasing its shadow and making itself nearly invisible. This relationship between the squid and the bacteria is mutualistic. The bacteria receive a safe environment and a steady supply of nutrients from the squid. The squid receives the ability to produce light, which protects it from predation and allows it to hunt more effectively. But the partnership is not permanent. Every morning, as the sun rises, the squid expels about 95% of the bacteria from its light organ. During the day, while the squid is buried in the sand, the remaining 5% of bacteria reproduce, multiplying until the light organ is full again by nightfall. This daily cycle ensures that the bacterial population stays healthy and active. The bacteria do not enter the light organ randomly. The light organ has ducts lined with mucus that is chemically attractive to Vibrio fischeri, but repels other bacterial species. When a newly hatched squid emerges from its egg, its light organ is sterile. Within hours, it begins to draw in water, filtering it through the ducts. Vibrio fischeri, which makes up only a tiny fraction of the bacteria in seawater, are selectively admitted. Once inside, they colonize the chambers, lose their flagella, and begin producing light. The squid's immune system recognizes the bacteria as beneficial and does not attack them. This symbiosis has been studied extensively because it provides a simple, tractable system for understanding how animals and bacteria interact. Researchers have learned that the bacteria influence the development of the squid's light organ. Without bacteria, the organ does not develop fully. The presence of Vibrio fischeri triggers changes in the squid's tissues, including the development of blood vessels and immune structures. This is an example of how symbiotic relationships can shape the biology of both partners. The Hawaiian bobtail squid is not the only animal that uses bacterial bioluminescence. Many deep-sea fish, including anglerfish, flashlight fish, and ponyfish, harbor bioluminescent bacteria in specialized organs. In each case, the bacteria provide light, and the host provides nutrients and protection. The bacteria belong to various species, mostly in the genera Vibrio, Photobacterium, and Alevibrio, and they have evolved independently in association with different hosts. Bacterial bioluminescence has even been found in some terrestrial organisms. Certain glowworms and railroad worms harbor bioluminescent bacteria, though these cases are less well studied than the marine examples. In every instance, the principle is the same. Bacteria produce light as part of their metabolism, and other organisms have learned to harness that light for their own purposes. The ocean, the forests, the night sky above a summer meadow, all these places hold their own forms of light, created not by fire or electricity, but by the chemistry of life itself. Bioluminescence is one of nature's most elegant solutions to the problem of communication, of defense, of survival in darkness. It has evolved independently across the tree of life, appearing in bacteria, fungi, protists, jellyfish, worms, insects, fish, and squid. Each lineage has found its own version of luciferin, its own luciferase, its own way of turning chemical energy into photons. The study of bioluminescence has given us more than just knowledge. It has given us tools. Green fluorescent protein, discovered in a jellyfish, now illuminates the inner workings of cells in laboratories around the world. Luciferases from fireflies and bacteria are used in assays to detect everything from contamination in food to the presence of disease markers in blood. The mechanisms that allow organisms to produce cold light have inspired research into new forms of lighting, more efficient and less wasteful than the incandescent bulbs and LEDs we use today. But bioluminescence is also fragile. Light pollution dims the flashes of fireflies. Habitat destruction eliminates the forests and wetlands where their larvae grow. Ocean acidification and warming may disrupt the delicate symbioses between squid and bacteria, between fish and their microbial partners. The bioluminescent bays of Puerto Rico, once so bright they were called liquid stars, are threatened by coastal development and pollution. The fungi that make forests glow are losing habitat as old growth trees are cut and replaced with younger managed stands. To preserve these living lights, we must preserve the places where they live. We must reduce light pollution, protect dark sky areas, and ensure that fireflies can find each other in the night. We must safeguard coastal waters, prevent nutrient runoff that causes toxic blooms, and protect the coral reefs and kelp forests where bioluminescent creatures make their homes. We must leave standing dead trees in our forests, the rotting logs where fungi grow and where firefly larvae hunt. These are small acts, but together they can keep the lights on. If you found this journey through the glowing world interesting, please consider liking this video and subscribing to Bedtime Biology. Leave a comment below if there are other topics you would like us to explore, and perhaps the next time you see a firefly flashing in the dusk, or walk past a rotting log in the forest, or stand at the edge of the ocean at night, you will pause for a moment and think about the chemistry taking place, the ancient dance of luciferin and luciferase, the light that living things make in the darkness. Sleep well.